Assalamualaikum warahmatullahi wabarakatuh. Hello, we are from Group B. We will present about nematode insect breeding and APH efficiency testing against insect. And before we start our presentation today, I will introduce our member. Next. And first, Johannes Mantanari, student number 5, Farika Pujiarini, student number 60, Amalsya Ikarohmi, student number 77, Sara Vanesya, student number 131, Gracela Nanda, student number 169, Rustiana, student number 192, Serina, student number 198, Desty Dwi Artanti, student number 233, and Isna Yuni, with student number 242. Next. Introduction. Pest control using pesticides has been considered as safe for the environment and human health in the long-term use. The intensive use of chemicals can use resistance and resurgence of the target pest. Hence, it may increase population level, which is higher than the initial population. One alternative method of pest control that is effective for insect and environmentally friendly is biological control using the formulation of specific microbes such as fungi, bacteria, and virus. Next. And then the entomopathogenic fungi such as Bevaria bassiana is a control agent that infects insect body by contact ingestion and widely distributed in nature in white house ray. Moreover, Bevaria bassiana, known to be safety to non-target organisms such as predator and parasitoid. Bevaria bassiana has been evaluated in the laboratory and applied in the field to control numbers of insect pests. Next.
Next is Stenerma sp. Stenerma sp is a type of nematode that can be used to control insect pests and other harmful insects. This is due to its ability to significantly suppress insect pest populations. The mechanism of NEP infection is mutualism with bacteria. In the absence of bacterial symbionts, nematodes can also kill house insects, but their production rate is low. Stenerma spp has several advantages as a biological control agent. Among others, it has high house-seeking ability, is selective for insects with a broad house spectrum. Next. Capybara borer or Hypotenemus hampei. Hypotenemus hampei, otherwise known as the capybara borer, is the most serious pest of cavy in many of the major cavy producing countries in the world. The life stages are egg, larva, pupa, and adult, and all are found inside the seed. Attack by Hypotenemus hampei begins at the apex of the cavy berry from about 8 weeks after flowering. During active female, she pushes out the debris, which forms a deposit over the hole. This deposit may be brown, gray, or green in color. Infestation is confirmed by cutting open the berry. Next. Hong Kong caterpillar or Tenebrium molitor. Tenebrium molitor RL or Hong Kong caterpillar is a warehouse pest that infests plant products and animal carcasses. These pests act as secondary pests and scavengers that infest grain products. These pests more specifically attack grain products whose physical form has been damaged or destroyed. Tenebrium molitor is a holometabola insect that is an insect that undergoes complete metamorphosis. The life cycle of Tenebrium molitor consists of 4 stages, namely egg, larva, cocoon or pupa, and adult insect or imago, which lasts for 3 until 4 months.
The next one is weaver ants or Oecophylla smaragdina. Weaver ants are one of the natural enemy of pests that attack plantation. They prey on a variety of pests, such as moths that are active at night and those that hide under leaves during the day. Weaver ants prefer protein over sugar. Protein can be found in meat, fish, chicken, mice, and insects. In addition to needing protein, weaver ants need additional food in the form of sugar. To get sugar, weaver ants prefer to look for sugar rivers such as honeydew, which is secreted by insects that suck plant fruits or nectar. The next one is Bevaria bassiana. Bevaria bassiana is a cosmopolitan entomopathogen, grows naturally in soil and is pathogenic to certain insect species. Bevaria bassiana has flat colonies, short hyphae, and round spores. Conidiospores have fertile parts that branch in a zigzag shape and form conidia that resemble balls at the ends. Bevaria bassiana produce a toxin that can cause aggressive paralysis of insects larvae and imago. Several toxins that have been isolated from Bevaria bassiana include bevaricin, bevarolide, isorolide, and diazoxalic acid. For the materials and tools that we use, the first one is nematode insect breeding. The materials are water, distilled water, alcohol 70%, Hong Kong caterpillar, chicken intestines, plastic wrap, aluminum foil. And the tools are petri dish large and small size, tweezers, Erlenmeyer, and autoclave.
Okay, the next is APH efficiency testing against insect. The material first aquades, Bevaria bassiana, coffee beans as a host, for coffee borers, Hong Kong caterpillars, ants. And the tools first trays, tweezers, cutters, plastic cups, and sprayer. Next. The practical implementation, first nematode insect breeding, first in vivo, culture directly on insect. The first one, prepare to breed petri dish in large and small. Next, put a little water on a large petri dish, put Hong Kong caterpillar or other types of caterpillars into a small petri dish, and then the next, fill up the two petri dishes and drop filling onto small water containing nematodes on the caterpillar's body. And then the next, cover with a large petri dish lid and make observation periodically on the second, third, fourth day until you see the caterpillar will dry and secrete. Already spring color will appear when and bacterial symbiont. Next. And the next, in vitro with chicken instant protein. First, prepare the intestines, wash and sterilize using 70 alcohol and distilled water, and then pure the intestines with distilled water until it becomes a thick liquid. And the next, enter the protein liquid from the intestine into the Erlenmeyer, cover with aluminum foil and plastic wrap, and then sterilize in an autoclave until it reaches a temperature of 121 Celsius. Then cool, put the protein liquid into the petri dish and put 3 drops of the liquid containing the nematodes into the same petri dish container. And then cover the petri dish, then wrap with plastic wrap. And make observation after 5 days under a microscope. Next. The next is practical implementation. Second, APH efficiency testing against insect. First, prepare the tools and materials in advance, and then look for 20 coffee borers pressed on the prepared coffee beans. Put the coffee borers into plastic cups and then put 180 Hong Kong caterpillars into 18 plastic cups and then spray Beauveria bassiana that has been diluted with aqueous into 9 plastic cups. Well, 9 plastic cups that were not sprayed become the control. And then the next is put 20 ends into 2 plastic cups. First plastic cup as a control and then others were with a spray of Beauveria bassiana.
Okay, the next step is insert the plastic cup containing Hong Kong caterpillar into the 3 trays that are already available, each containing 6 plastic cups. The trays were divided into 3 counter samples and 3 samples treated with Beauveria bassiana. After that, distinguish the coffee border and plastic cups into control sample and samples with treatment. And then the last step is make observation 4 times in a row. Next, we are going to talk about the observation result and discussion. Spore density. The density of spore per millimeter of water is one of the factors successful control of pests and disease using biological agent. Calculating of spore density and Beauveria bassiana done for determining the number of fungal spores produced. In calculation, these spores were carried out using a now better improved hemocytometer. Next. There are several ways that we can do to test the spore density. The first one is weigh 1 gram of Beauveria bassiana and then put in an Erlenmeyer and add 100 milliliter of sterile distilled water and Erlenmeyer closed using aluminum foil. And then shake the suspension for 3 minutes. After that, take the suspension as much as 0.1 milliliter using pipette and pour into a canal filled of the hemocytometer until the canal completely filled with the suspension solution. After that, let's stand for 1 minute so that can be stable. And observe the spores of Beauveria bassiana using microscope. And then the next step is calculation of spore density is carried out on the A, ABCD, and B, ABCD. And then observation were made with a magnification of 40 times and calculate manually.
And this is our table result of calculation of spore density Beauveria bassiana. There are 3 repetitions, and the first repetition, there are number of spores per milliliter, 9.425 multiple 10 to the 7 power. And the second repetition, there are 10 multiple 10 to the 7 power. And then the third for 5 multiple 7 multiple 10 to the 7 power, and then the average of the 3 repetition are 9.625 multiple 10 to the 7 power. Next. And then this is table of Beauveria bassiana treatment of Hong Kong caterpillar. There are 3 samples with the 4 observation time on 7 June, 8 June, 10 June, and the last 11 June. Hong Kong caterpillar as control in samples 1, 2, and 3 didn't experience significant mortality. Meanwhile, the Hong Kong caterpillar that were treated with Beauveria bassiana died every day. Next. And then this is table of Beauveria bassiana treatment of coffee borer with the 1 sample and the observation time SEMS of the and the treatment of Beauveria bassiana against the coffee borer. The same thing happened in sample 1, 2, and 3 didn't experience significant mortality even from the beginning of the observation. The coffee borer treated with Beauveria bassiana suffered a total death. Next.
Okay, the last is research about Beauveria bassiana's treatments of ants. While on the ants, on the first day of observations, there were still one alive, although after that, all the ants also died in Bouveria bassiana treatment. And the mortality of control on the first day is 7%, second day is 90%, and then 100% on the next observations. Next. Based on the observations, the mortality rate of insect with Bouveria bassiana treatment was higher than the control treatment. This happened because the insect were infected with Bouveria bassiana, which is an entomopathogenic fungus. So their appetite was reduced. The caterpillar become less active, stiffened, hardened, and died. This is accordance with Wahyudi 2002, which states that the toxic product by Bouveria bassiana include bouverizins, which can destroy the fat layers and increase permeability, which can destroy specific ions so that it can cause abnormal ion transport and then damage the functions of cell or organelles of larva cells. Next. The density of spores per millimeter of water is one of the factors successful control of pests and disease using biological agents. The densities of spore Bouveria bassiana tested show a result of 9.625 across uh, 10. This is a value is the higher than the exciting standards according to the Directorate of the Plantations Protection Ministry of Agriculture 2014. Good density of the sport for fungi Bouveria bassiana is 1 across 106 spores per gram. This is in accordance with Prayogo et al. 2005, which state the mortality of insect is very determined by the density of pathogenic fungus. Of the pathogenic fungal conveners applied and higher, the conidia of higher the mortality. With a low density of fungal spore, fungi still take time to infect insects until it causes symptoms of infection, even though contact between the two has occurred. The higher the density of fungal spore Bouveria bassianus, you that has been applied to feed or plants is able to the poison test insect. Next.
Then, on the efficacy test on ants and coffee borers show that many insects on the treatment of control are dead. This is due to the poor initial state of the ants, and also the container for laying ants is small and there are just small air holes so that the air circulation becomes poor, which result is in the ants dying due to the high level of carbon dioxide in the container. While coffee borer die can because at the endosperm is not available substrate needed or can be caused by lack of oxygen availability for coffee borer. The less amount of air hole will cause poor air circulation and make the oxygen supply will be depleted. If it is exhausted, then insect will suffocate due to lack of oxygen in the blood. Insects will die in the oxygen concentration in the air drops by up to 2%. Next. So based on our discussion and the activity that have been carried out, it can be concluded that the spore density of Bouveria bassiana, the result is 9.625 multiple 10 7 milliliter, so that the fungus have met the against quality standards biological. Spores produced by Bacteria bassiana, which is reproduced using milled corn media, has good growth. Treatment by applying Bacteria bassiana to caterpillar pests, Hong Kong, Hampei, and Weaver, and show their effectiveness to infect the pest compared to controls. Next. Okay, that's all from our discussion today. Thank you for watching, and wassalamualaikum warahmatullahi wabarakatuh.